Good morning, good afternoon. Thank you for being here just before lunch, and thank you, ARM, for inviting us to speak in this beautiful city of Rome. My name is Stefan Ehren, and I'm talking to you today about BlueRock Therapeutics. So just at a high level, I'll briefly walk you through some of the exciting data that's coming out of in the cell therapy space and how we feel this is going to translate well into the therapies that we're developing, going out of oncology into other degenerative diseases with high unmet clinical need. And what we have done at BlueRock, to what Stefan has just said, developing this entire pipeline of generating iPS cells and derived products, but then also building the capabilities to do clinical translation of this work. And I'll close that with showing you some of the early data from our trial in photoreceptors that we developed together with Opsys, and of course our most advanced program, bemdaneprocel, that's in the clinic in phase 1, and we read out the 18-month data there. As many of you have seen graphs like this over the last 1.5 days and before, this is really an emerging market where we see a number of approvals, not only in the CAR T-cell space, but also now in the engineered and non-engineered autologous and allogeneic cell therapy space. And a lot of the development that groups like CDMOs and other clinical developers and therapeutic companies are putting into this to advance these therapies to the patient that so desperately wait for them. And we've learned a lot about manufacturing capabilities, regulatory interactions with payers, manufacturing part. And that leads to a large series of clinical trials that are currently studying cell therapies across the phases of clinical development, leading all the way to phase 3 and approvals. And so we're expecting that by the end of 2026, we'll have about a $25 billion market here. BlueRock itself, we focus on pluripotent stem cells and induced pluripotent stem cells. In most of our programs use the iPS cell platform, where we start with a highly characterized individual donor that's healthy and completely screened for absence of diseases. We turn those cells into pluripotent stem cells, which effectively refer to as a blank canvas cell that we can turn into any cell type of the body. And they can be mass-produced, banked, and serve an indefinite source of our therapeutic products. And in the second arm of the company, we apply genome engineering using CRISPR-Cas enzymes to modify the genome to enhance the therapeutic potential of the cells. And together, that's a really powerful tool, because now you can, in theory, generate any cell type of the human body and engineer any gene you want into those cells, and do so at scale. Our approach, in contrast to some of the others you have seen at this conference, is an allogeneic approach, where we take, as I said, a healthy donor, bank those cells and mass produce them for application to the patient. That obviously has implications of cost of goods, but also you have to think about how some of the autologous cell therapies suffer from the delay it takes from collection of donor material to the application of the patient. We effectively have a cryopreserved product that's ready overnight to be given to a patient. So in cases where the disease is very grave, you obviously gain that time. We pride ourselves in having founded all the work that we do on excellent science, and I think that really helps us to translate the findings from the lab into the clinic and see the data that we're starting to see in our clinical trials. That's around Lawrence Studer and Vivian Tabar on the Parkinson side, Mike LaFlamme and Gordon Keller on the cardiac side, and Bruce Blazer in our immunology programs. I've get asked this question here a lot, how do we work with Bayer? So BlueRock was founded at the tail end of 2016 through a joint venture between Versant and Leaps by Bayer. 2 years into this venture, Bayer decided they would acquire us outright. So for the last 5 years, we've been operating as an independently operating company under the Bayer umbrella. It's referred to as an arm's length company, and it's really actually works really well for us, and hopefully for Bayer as well. We were the poster child arm's length company. They subsequently acquired AskBio, a gene therapy company, building Bayer's cell and gene therapy platform. Over the last 7 years, we've built the pipeline that Stefan just described in a CDMO model internally, translating academic findings into clinically meaningful clinical trials. including not just scaling up and understanding the biology better, but also building the capabilities of turning a flask-size differentiation into a bioreactor-size differentiation to ultimately meet the clinical demand that we're expecting for some of these therapies that we're developing. And we had to build a lot of those technologies and also the people and staff that simply wasn't in existence a couple of years back. And that included dedicated clinical development and regulatory teams that can help us push these therapies through the regulatory programs. And importantly, we're able to build an in-house facility that allows us to manufacture our therapies for phase 1 clinical trials, and through the relationship with Bayer, leveraging their Berkeley Biologics campus, tech transferring that to that site for commercial manufacturing. And today we are in the tech transfer process from our Parkinson's program to manufacture that at scale in a commercial setting in Berkeley. BlueRock as a company is focused on 4 therapeutic areas: neurology, cardiology, ophthalmology, and immunology. And our lead programs are listed below. I'll talk more about OPCT01, our ophthalmology program that we developed together with Opsys, and our in-house neurology program called the bemdaneprocel. I'm happy to talk about our cardiac and myeloid program later if you find me during lunch. So the OPCT01 photoreceptor product is quite interesting. If you know anything about inherited retinal diseases, it's caused by a variety of genomic defects. There's about 70 known, and a large proportion is unknown. They all lead to either the failure of the photoreceptors ever to develop or for them to degenerate over time. And we've seen great success with gene therapy, such as Luxturna, developed by Spark and now owned by Novartis, that targets one specific mutation. If you think about gene therapy, has to then always be tailor-made to that specific mutation, if you even know it. The approach that we're taking is making photoreceptors that contain a full set of healthy genes, and in theory can then be used to treat any of the underlying genetic causes, because you're actively replacing the entire photoreceptor with its full capabilities. So we believe we can target a large market of those patients, about 200,000 patients that are currently affected by diseases that would fall under this umbrella. Obviously, again, that reduces the cost of manufacturing for us and allows us to develop these therapies for a broader number of patients and giving them access. Some of them are really only in the small percentage range and would probably otherwise never see a gene therapy. We're planning to file an IND this year and start treating patients at the tail end of 2020. This just summarizes the work we're doing here. We're making photoreceptors one of the 2 main cell types in the eye, together with RPE. Our collaboration with Opsys allows us to develop both cell types, and they're surgically transplanted in the back of the eye, and then under immunosuppressants engraft, and turn light signal into electrical signal that the brain can interpret as pictures and motions. And the idea is that we can again, make a cryopreserved product, bring this to the patient in need, transplant them, and then upon engraftment, restore their vision. We're still developing the additional program with them using photoreceptors and RPE cells together, which would open the market to patients with age-related macular degeneration. But I'm going to talk a little bit more about Parkinson's. I think everybody I've spoken here at the conference has a friend, family member, or colleague that's suffering from this disease. It's really prevalent. It's the second most neurodegenerative disease in the world. It affects about 8.5 million people worldwide. And the neuropathological hallmark is the loss of dopaminergic neurons in the substantia nigra. So most therapies today are targeted at helping the body cope with these symptoms. None of them are aimed to restore the function of these lost neurons. And so what you see is permutations of levodopa therapy that's been around since the early 70s, such as levodopa-carbidopa combinations or pump formulations that help the patient to get a more sustained release of levodopa that then in the brain requires dopaminergic neurons to be converted to dopamine to have the function. And over time, as more and more neurons die, the functionality of levodopa, carbidopa, and permutations thereof decrease. And then just 2 other technologies, deep brain stimulation and focused ultrasound, and really targeting the side effects or the main motor problems of the patient by giving electrical stimuli or ablating parts of the brain that cause these symptoms. overall not a satisfying solution. So that's why we set out to develop bemdaneprocel, or DA01 as we called it earlier. This was run in a phase 1 open label, multi-center, multi-site, non-randomized study that we now refer to as EXPEDITE, and the drug product is bemdaneprocel. It was aimed to improve health-related quality of life in patients with Parkinson's disease. Today we're reading out our 18-month data, which for us is significant because it is 6 months after discontinuation of immunosuppressants, suggesting that if successful, and I show you the data, that the graft is stable for at least 18 months, even in the absence of lifelong immunosuppressants. The trial design was quite simple for EXPEDITE, very traditional. We enrolled in both Canada and the US, slightly different enrollment criteria based on regulatory feedback in patients with moderate Parkinson's disease. We used fluorodopa PET as an imaging tracer to trace persistence of the cell and engraftment of the cells at baseline 12-18 months, and we plan to do so at the final study endpoint of 24 months. And we followed it, of course, with clinical endpoints at the matching time points as well. 2 dose cohorts were enrolled, both of them targeted to be therapeutic, and we called it a low and a high dose. Was done in a single surgical session. And then, as I said, 12 months immunosuppression and the matching follow-ups at 18 and 24 months, which we'll reach this year. The surgical procedure required 2 burr holes at the top of the head, access to both hemispheres. Along this one burr hole, we made 3 needle tracts and 3 deposits at 3 different depth levels for a total of 9 deposits per hemisphere, or 18 total. We gave concurrent immunosuppressants for 1 year, as I had mentioned. It's listed on the left side of the slide. I should say, we're pretty excited about the safety data from this trial. We had only 2 serious adverse events not related to the drug itself. They were surgery related, one-time seizure, and another person completely independent had a fall. Everything else was not serious and was related to the immunosuppressants, and after we discontinued that, a lot of those signals came down. If you look at UPDRS scores, a clinician-administered rating scale, you can see here the high and low dose in light blue and dark blue, and what the graphs show is the median mean and the extremes. You can see in particular it's almost a dose-dependent effect. At the high dose over the course of 18 months, the far right in the graph, all patients significantly improved the motor rating administered by the physician. They started at about a score of 50, which is really debilitating for patients with Parkinson's, and reduced that down to 23, so almost a 50% reduction. And to see that over the course of the disease, or the treatment, that's expected because the cells do need to engraft and make new connections to get their functionality. We actually can expect this trend to continue in a positive way. This matches what the patient sees, so that we do a Hauser diary where the patient, over a couple days leading up to the exam, writes a diary for 16 hours of the day. They say for every 30 minutes how they're feeling, and they say they're either good or bad, which is referred to as on or off time. So these patients start with the high-dose patients with 11 hours of on time, and they report at 18 months an average improvement of 2.7 hours. So they're almost reaching 16 hours of good on time, which is remarkable. And I'm not showing the data, but that matches the off time here as well. So they see a corresponding reduction in the off time of the symptoms. Most importantly for us, we did PET imaging, and you can see here at the bottom, a normal Parkinson's patient, you see that sort of C-shaped or banana-shaped red signal in a healthy patient that deteriorates from the posterior side of the putamen. The signal gets smaller, and when you look at the patient composite image of all the patients here, at baseline, you can see them correctly diagnosed as moderate stage Parkinson's patient, where you see only sort of a small red blob in the PET signal. That becomes bigger as we engraft cells after 12 months, and then it becomes even slightly larger, and it definitely is persistent at 18 months, again, after discontinuation of the immunosuppressants, showing us that we can really administer this therapy with a short period of immunosuppressants. So, as I said, bemdaneprocel was well tolerated with no serious adverse events related to the to the drug product. Cells engrafted and survived, and we have encouraging exploratory clinical endpoints that lead us to develop this into a phase 2 trial that we begin to enroll later half of this year. So with that, thank you very much. Excited to have any questions over lunch.